So Ruaridh is actually here having won the right to stand in front of us, and he got some free pizza as well. The grad school conference, the one that's last week in January or early February, whatever it is, which is organised by the grad school, and is basically a sort of unofficial conference run by the PhD students for the PhD students here. They have sort of competitions to see who's got the best talk, and Rory had the best geological based talk, didn't you? So congratulations, this is your prize. I hope you enjoy it.
Is that on? Yes, I think it is. Great, thank you very much. So I'm going to talk about some fossils from the Ediacaran Cambrian transition, which is this period in Earth history during which we see the emergence and then diversification of animal fossils in the fossil record and the establishment of modern day groups in the Cambrian. And I'm going to focus on one particular organism from the latest Ediacaran, and that is this one here, Namacalathus. But first of all, I'll give a little bit of background as to why we're interested in these Ediacaran organisms. And here's our time scale here. So we have the Ediacaran and Cambrian, and the boundary is at about 539 million years ago at the moment. That changes sometimes. But in the Cambrian, we start to see the emergence of fossil groups and fossils that we could correspond to groups that we would recognize today. So things like these preapulid worms at the top row here, these trilobites, which are obviously arthropods, and these mollusks here. And all of these are very recognizable to us, and we could put them into pretty definitive groups by ourselves. But in the Ediacaran, we still have a really diverse fossil assemblage, but all of these things look quite weird, and they're quite difficult to ascribe to particular modern day groups. And the key to sort of understanding early animal diversification in the Cambrian is understanding how these Ediacaran forms relate to forms that then appear in the Cambrian. So the first Ediacaran organisms that are widely considered to be animals are these sort of frondose taxa that look a bit like leaves here, that appeared about 575 million years ago. The oldest ones are in Newfoundland and Canada. But the first animals that start to biomineralize, and that's produce a calcium carbonate exoskeleton, like these guys up here, appear at the same time at about 550 million years ago, so right towards the end of the Ediacaran. And Namacalathus is one of these organisms that we're interested in here. And one place that we might go to try and find Namacalathus is the Nama group in Namibia, which is a primarily carbonate group, although with some siliciclastic successions as well, that dates from about 550 to 539 million years ago, so right up to the Precambrian-Cambrian boundary. And this is from a place called Omkek Farm, and we have a lovely carbonate succession there in the background. And at this point in time, Namibia on the Kalahari Craton in red here is sort of tropical, which is why we have these carbonate buildups. But Namacalathus is not only found in Namibia, but also in a number of geographically disparate places, such as Laurentia, so they're found in Canada today, Oman, and on the Amazonian Craton, so there's some in Paraguay. But what exactly is Namacalathus and what does it look like? So as I said already, Namacalathus is one of these skeletal animals, and it's obviously one of the first animals that we see in the fossil record. And it's composed of this sort of broadly spherical cup-shaped structure that has a central opening right at the top that it would probably use to feed or something similar to that. It has these openings right around the side of the cup that we call lumens, and then that sits on top of a stem. And Namacalathus is really efficient at colonizing bedding planes. So this is just a slab, and all this dark material is just a whole load of Namacalathus on the slab. And they are one of the organisms that form the first animal reefs, some of which you can find in places like Namibia in the fossil record. And it was first described in 2000, the year after I was born. And it was reconstructions based on a lot of these fossils show something that looks a bit like this. And we have this, cup with central opening, these lumens around the side, and this stalk here. So one of the keys to sort of understanding what Namacalathus might be is looking for soft tissue preservation, because this often informs us of the possible phylogenetic affinities of an organism, i.e. where it places on the tree of life. And this is a place called Svartmodder Farm in Namibia. And this is a carbonate succession. And a few years ago, the research group here from Edinburgh, Rachel Wood with some of her PhD students, went out, and Amy Shore, who was a former PhD student there, is carrying something in her right hand there that you can maybe just about see, and that is this slab here. Now, this slab has a lot of Namacalathus dotted all over the surface, and we can see them here, and it's also got these elongate cloudinids, which are other animals. But these Namacalathus have a particular feature that is of interest, and that is that they have this sort of orange crust on the outside. And this is iron oxide, which has replaced pyrite, but retained the original pyrite rhomboidal structure. And it's really early pyritization of soft tissue. So this provides us an idea of some new features that we can sort of get at. And the thing about it being iron oxides within a carbonate rock is that there's a nice density contrast between the two materials, which means you can CT scan them. And when you CT scan these fossils, you get something that looks a bit like this. So we have the cup structure that remains. So this is all this blue material, soft tissue, and that's lining the cup there. And then we have all of this. This is the crust that we saw on the top, and we call these lobes, and they sort of form around the central opening. But curiously, when you take sections through Namacalathus, it was found that we have this interesting little internal structure here labeled CT. And this was tracked throughout a number of sections in individuals. And it was shown that it was broadly J-shaped. And it was suggested that this is representative of partial through gut structure. Now, if it has a through gut structure, it means that Namacalathus is a bilaterian, so related to us in some way, has bilateral symmetry when it develops. And this goes against initial interpretations, which suggested it was a cnidarian, like corals, anemones, jellyfish. So this points to a slightly more derived placement for Namacalathus within the tree of life, rather than its basal position within the cnidarians. So I'm going to show you a few more reconstructions that I've done of some other individuals from the same slab, and what we can get from information we can get from these. So the first one here, the density contrast between the skeleton and the surrounding sediment was actually good enough in the CT scans that we were able to pick out the skeleton itself, and not only 3D reconstruct the soft tissue here in browns and golds, but also the skeleton in green. And this skeleton is populated with a number of really large spines, and we can see them here on the right as well. So they often occur in pairs that curve towards each other, or single pairs that sort of hook around on their own. And it may be that that Namacalathus is using this as an anti-predation mechanism. What's preying on Namacalathus, we don't know. But it might also be that it's using it to anchor itself to a substrate. And curiously, none of these specimens have stems. They are all directly attached via a basal opening to the microbial mat that they were living on. And we see here that we have the Namacalathus sort of sitting inside the cup there. And this is the first reconstruction that we've been able to make of a Namacalathus skeleton with the soft tissue of this kind. So it's quite nice to see. But if we look at another individual and look at the reconstructed CT data, we can see that this red material here traces this crust quite well. These are the lobes, but we have this green material, and this actually penetrates into the cup itself. And when we take some sections through this specimen, we can see that, sure enough, we've got this high-density iron oxide orange material that we can trace through multiple sections, a whopping 0.6 of a millimeter in thickness, which is quite big given the size of Namacalathus. It's only about 1 centimeter in diameter. And we think that this might be another example of this possible gut structure. And when you put all of these images together and reconstruct them, you can see that we have these, purple lobes that correspond to the lobes here. But then we have this basically continuous green central tube-like structure that almost connects to the other end there. So we think this is a slightly more complete version of the thing we're seeing in the previous slides, and it might be more evidence for a sort of bilaterian through gut and bilaterian type affinity. And the final specimen that I want to show you, we'll just look at this reconstructed CT image first. We can see all of this really high density material. And again, these are our lobes. This is the central opening. But interestingly, we have these two discontinuous layers of high density material. And these are representative of a layer of tissue on the inside of the skeleton and on the outside of the skeleton. So the skeleton's in there, but the density contrast isn't good enough for us to see it. So when we reconstruct that, it looks a lot like the reconstruction we saw before, but the difference being here that this is all soft tissue. So there's no skeleton here that we can see, just soft tissue. And one of the striking things is that we can still see the lumens, but they're really depressed. So we have this layer of soft tissue covering the lumens themselves. So rather than it being open into the body cavity like we see in the non soft tissue preserved fossils, we actually have a layer covering that. And that may be because it's using it for something like asexual reproduction. And sure enough, when you look at the association of other specimens with each other, we can see that we've got a top-down view of a Namacalathus here. So we're looking basically down into the central opening. And we have these lumens around the outside here. And this is the basal opening of another Namacalathus that is basically in really close association with the lumen of this one. And then we see the same thing again here, top-down view into the cup, and then basal opening, and then the cup there. So it looks like these lumens are being used as brood chambers from which cells can divide from this tissue and form a little bud, and then another little Namacalathus can grow and then separate from the parent individual at some point. And then these can sort of colonize a surface and form our first animal reefs. So we put all of this together, we get something that looks a bit different to the orange 3D reconstructions we saw at the beginning. We have this skeleton. Now, not all Namacalathus skeletons have spines, but these ones have whopping great spines. And we now know sort of what the animal looks like inside. So we have this layer of tissue going right along the inside of the skeleton, then these lobes at the top that were possibly able to expand and contract as the organism needed to. Have tissue covering the lumen like a brood chamber there. And we have a layer of tissue on the outside. And this is quite important because it might be similar to the layer of tissue that a brachiopod has on its skeleton, which is called a periostracum, and it's used to prevent corrosion on the surface. And if that's the case, then it may mean that Namacalathus is related in some way to brachiopods and an ancestor to them, and sort of further fits the bilaterian affinity. And finally, we've got more evidence for this central U-shaped structure, which is possibly a through gut, which would once again furthermore point towards bilaterians, which contain all sorts of groups: arthropods, nematodes, mollusks, and obviously chordates like us. So on that note, I'll leave you with a final image of Svartmodder Farm, the former stomping ground, as it were, of Namacalathus some 540 million years ago.